to the BioHub podcast. Our guest today is Jason Bock, CEO of CTMC and a trailblazer in biotech innovation. With over 20 years of experience, Jason has led the evolution of monoclonal antibodies from novel concepts to mainstream therapies and is now driving breakthroughs in cell therapy. He's worked at companies like Teva, MD Anderson, and is now tackling a complex challenge over at CTMC. So thank you so much for joining us, Jason. How are you today?
Very good. Thanks for having me on, Rebecca.
Yeah, absolutely. I'm very excited to have you on here today. So yeah, just to obviously get straight into it. We've obviously spoken before, Jason. Had a great conversation. Your background is obviously extremely impressive. So I guess to start with, what initially drew you to the complex world of biologics and cell therapy?
Overall, it was that there was tremendous value in these therapies, these kind of modalities, being able to address unmet medical needs. And yet, in both of these fields early on, there have been a lot of challenges associated with being able to develop, manufacture them at a reasonable scale and cost. And so there was this great opportunity or challenge in if we could figure out more robust ways of producing these, quicker ways of getting these products into clinical trials, more scalable ways of manufacturing them for commercialization, then we could help a lot of people. And that's been the driving force throughout my career.
Yeah, absolutely. So I know obviously the cell therapy aspect to your career is a bit more new. You've been a very early pioneer with monoclonal antibodies. So how have you seen the industries, you've been there right from the beginning, which is awesome. So how have you seen the industry's approach to these therapies shift over time?
Yeah, so, it's been an interesting ride for sure in protein therapeutics overall and monoclonal antibodies specifically over the last 20 or 30 years. Developing therapies for patients and these new modalities, as much as we have urgency around getting them to patients as quickly as we can, these are really complicated things we're trying to do, and oftentimes does take decades for fields to mature. And so I've sort of had that perspective sort of growing along with the antibody field, where in the 90s, they were not huge commercial successes, and it was not very easy in the beginning. And we faced many, many questions about the viability even of producing either protein therapeutics or antibodies relative to what dominated therapeutics at the time was small molecules. And yet these proteins had such exquisite activity, or in case of monoclonal antibodies, exquisite precision, that we knew if we could harness this and mature this modality, they would be a big therapeutic benefit. And so, it was from quite humble beginnings when we would produce these, we would produce them at very low titers, less than 100 micrograms per liter of culture. When we would test these, we would run a STS page gel and stain it with Kumasi and look at it and say, okay, it looks like about one band, so it must be pretty pure. and over 20, 25 years, we've come so far in being able to detect with, let's say, advanced mass spectrometry, one Dalton differences and very with high sensitivity. And in addition to the deeper analytic understanding how those analytics or those quality attributes related to therapeutic efficacy and or safety. Mm-hmm. And so that's like what a mature modality looks like. Hey, we can produce these at high quantities, at relatively lower costs. We can test them so that we really understand if there are any batch-to-batch differences. And if there are those differences, we understand the clinical implications of those. So that's the maturation that the monoclonal antibody field has gone through. And as I see it, cell therapy is in a lot of ways at a very similar stage that antibodies were 25 years ago. And that I firmly believe we will be able to mature this field and address those same challenges. My hope is we can find shortcuts and quicker ways to do that, so it doesn't take us 30 years. But right now, whenever you're sort of looking up such a steep hill, the mountain looks really high. So I know there's a lot of challenges in front of us. We just have to keep taking one step at a time and being very intentional with how we overall try to mature this field.
Yeah, no, absolutely. Because obviously the cell therapy space as well is, really starting to get really interesting. But obviously the treatment itself that are successful are extremely expensive. So it's not as easily accessible for everyone. So hoping that we can replicate what has happened with monoclonal antibodies into cell therapy.
It's interesting, Rebecca. I think one of the things that's happened in cell therapy field that was a little different than monoclonal antibodies is that, the first CAR Ts that were commercialized were very effective. Like you're, curing cancer of patients who had no other options and then didn't get another treatment for the rest of their lives, with a single dose. That wasn't the case with early antibodies that were not all that efficacious when we started with chimeric antibodies that had some immunogenicity issues and were not the greatest, didn't have the greatest therapeutic profile. And so I think in some ways the field of cell therapy is under more of a spotlight at its early days than the field of monoclonal antibodies was, because these products are so efficacious and have the potential to help so many patients. There's even more pressure on us to be able to find ways of rapidly making them scalable and reducing the costs.
Yeah, absolutely. So when you first encountered CAR T therapies, what did you think stood out as probably the biggest challenge in translating this groundbreaking science actually into a viable treatment?
Yeah, these are clearly the most complicated therapies we've ever tried to develop as humans. They're living cells that we're engineering to do something specific, but are many orders of magnitude more complicated than antibodies. And that was the other interesting parallel. For 20 years, we would say, these antibodies are tremendously more complicated than small molecules. And we would compare it like, a bike would be a small molecule and like an airplane would be an antibody. I think if I haven't tried to carry out that analogy, but I think if we carry that out to cell therapies, I don't know, it would be like aircraft carrier or something that you're trying to create and engineer. Just a lot of moving parts. They're living cells that change over time. That you're trying to engineer and control those processes to produce those.
Absolutely. Amazing. So I guess I touched on it in the beginning here. Obviously, you've worked at some amazing companies, working at companies like Teva, moving to MD Anderson. When you moved from Teva, you were obviously leading a 600-person organization. And then when you moved over to MD Anderson as the first employee in a new group, how did that shift typically either affect or really utilize your leadership approach?
Yeah, it's interesting, Rebecca. When you put it that way, it seems like a very different situation. But in many ways, I saw some parallels. So the way I came into Teva Pharmaceuticals, which is the world's largest, best known as the world's largest small molecule generics company, one time had over 50,000 people. And they acquired the small private company I was in that was about 30 or 40 people at the time. And really I always positioned us as like a small company within a big company. And so we were doing innovative biologics within a large generics framework. And over time, we were able to build more and more capabilities on the innovative biologics side, until by the time I left, the CEO was quoted in the Wall Street Journal as saying, innovative biologics is the growth driver for Teva going forward, which I was very proud of, that we came from a small group, to have an impact on the organization. And in many ways, it was a similar kind of thing. So I came in to MD Anderson, and with the opportunity to build an internal biotech focused on cell therapies within the #1 cancer center in the world. Yeah. So a 27,000 person organization that does a wide variety of different things, but all at the end focused on ending cancer. That's like MD Anderson's very, very clear and focused mission is to end cancer. And starting a group within MD Anderson that was very different than what a lot of the rest of the organization was doing, had challenges and a lot of opportunities. So in some ways it was, the group within MD Anderson there called Therapeutics Discovery, this internal biotech, was like a small company within a big company.
Perfect. Understood. And obviously that's now translating to your CEO role over at CTMC, which is like a spin out from MD Anderson and with resilience as well. So you've got quite a unique model that helps biotechs progress from research to proof of concept. So what makes this model so effective for early stage companies? How are you so successful?
So it's been a fantastic experience, working with the people at MD Anderson to grow the group. Had amazing colleagues there to work with who were doing similar things with small molecules as well as antibodies. And we had the chance, because we were starting something new in cell therapies, to really build something fit for purpose and look at the challenges that companies were facing in developing new cell therapies and come up with something that we could do differently that would add value, right? And so MD Anderson had this, amazing clinical enterprise where more clinical studies in cell therapies are run at MD Anderson than any other place in the world. Tremendous patient volumes coming through. And how could we create an organization that would complement that amazing characteristic of all of this clinical capability? And with cell therapies, since for autologous cell therapies, all of the batches start with the patient. So, we start with the patient, either blood or tumor, and then produce a batch of cell therapies, and then we turn that to the patient. So you have this very patient adjacent manufacturing process that benefits greatly from manufacturing being adjacent to the patients. And so we thought that could be a differentiating characteristic for us, and that if we could create an end-to-end solution for partners where they have an innovative product but then need to manufacture it, run a clinical study with this very integrated manufacturing, that we could provide a real fit for purpose solution to that. So in a nutshell, that's what there's, I think it's always a good general premise. If there's something that's really hard for other people to do, and you can find a way of making that easier, that you're, in a situation where you can do that easier than they can do it, that's where you can add value to them. And so that's the very underlying principle of how can we add value to partners' therapeutic development programs. And it's worked out pretty well. We've filed 8 INDs on different cell therapies over the last 2 years. We've had good, 100% success rate thus far in getting those INDs, cleared by FDA and starting clinical studies, and have enrolled a lot of patients on those clinical studies in a very efficient and overall cost-effective way for our partners. So we've been able in some ways to sort of crack this nut of being able to do things overall in a more capital efficient way while still reducing risk, because there's not as many handoffs between partners, and we're pretty good at doing things. So we've been able to reduce risk and actually do things quicker.
Yeah.
It's very rare when you can get all those things combined. Usually there's trade-offs on those, but we've been able to really deliver all 3 of those on a number of projects over the last several years. And I think if you do that, if you have a model where you can add value, you can deliver these things that are really hard to do in other places or other ways. Lots of times if you mix those things together, you'll have success.
Yeah, no, 100%. And that is definitely success to be proud of. I can see why you are so happy with that. And 100% success, you don't really hear that number as much anymore. So that's always...
I should find some wood, make sure, we have ongoing submissions. So, but, one of my other phrases that I like to say is, people say, oh, you could be lucky. Luck is where opportunity meets preparation. And so, I think we've thought a lot about how to design the group. We've been very intentional about how to build the group, how we partner with biotech companies. So I think we've done a lot of the right things to prepare. And then when we have these great opportunities to work with amazing biotech companies and help them develop their products, we just look to take advantage of that opportunity.
Yeah, definitely. And I know that we've spoken in the past, but when we have spoken, you mentioned about it's important for leaders to leverage past experience, but just not necessarily repeating it. So it's using that experience with that. You've done it well at CTMC, but how have you done that at CTMC? Because obviously, they are different. But like you've just said, you've got 100% success rates so far. So how have you managed to leverage that?
I like that, Rebecca, that you're keeping us honest. 100% success rate so far. Let's see how long we can keep it up. But so I really do think it is important to leverage your experience, learn from that, learn from your successes, learn from your failures. Although I think it's very common to say you learn more from your failures than your successes. And I think there's some truth to that. On the other hand, if you fail, you're like, okay, something didn't work. Let's think about why it didn't work and try and do something different in the future. If something did work, though, and you were successful, I think being equally as thoughtful about why you were successful and understanding not just what you did to be successful, but why you were successful or how you did something that made you successful. And if you go through that process of leveraging your experiences, then you're in a great position to apply them in the future, as opposed to just like, oh, this is the way I always did in the past. This is the way I always built groups in the past. We're going to build it the exact same way here. I think one of the things I enjoy the most is building teams and putting amazing groups of teams together. The way we built the group at Teva was very different than the way we built the group at MD Anderson and at CTMC. There are some underlying similarities, but there are some differences as well. At Teva, we were trying to build a team that could robustly commercialize antibodies. And so we were very intentional in finding employees who had done that before at large, successful pharmaceutical companies like, Amgen, Genentech, Merck, BMS, and pulling them over with the lens of people who really understood why they had been successful. Not just what I didn't want to do is like, oh, we're going to make antibodies the same way Amgen does. No, we came up with a way of we're going to make antibodies the Teva way. But the Teva way leveraged some learnings from various other successes that other people had had. Now, the cell therapy field was much less mature than antibodies, and so there weren't as many successful, let's say, companies to pull people from. However, where there was a depth of cell therapy experience was actually in academic centers where people had been working on cell therapies for decades, both manufacturing them in academic GMP labs, as well as research labs that in some ways were doing, even though they wouldn't call it this at the time, but kinds of process or analytical development. And so we intentionally built CTMC with a mix of people with more academic depth of scientific background, some assistant or associate professors or advanced research assistants coming over to the group, combined with people with more traditional therapeutics development experience. And I have to say, at first, that mix was a bit of a clash of cultures and was a little difficult to manage through. But those guys really, everyone jumped in with both feet, learned from each other, and now we have this very integrated group that has this hybrid background of a depth of academic experience as well as a breadth of industrial understanding of therapeutics development. And I think that's an underlying secret to our success. So there's an example of building teams is really important. And so, I've done it a number of times in the past, but we were very intentional in building the team a different way at MD Anderson and in CTMC than we did previously at Teva.
Yeah. And I think that's really interesting that you say about learning from successes and failures, because I think so many people in every kind of walk of life, no matter what you're doing, always say learn from your failures. Yeah. Learn from what's been done wrong and do it a different way. But I think that's so right in saying to learn from your successes, because if something's been done correctly, then it can always be modified or changed for a different area of where you're trying to do the work. So I think I've actually never really heard that before, to learn from your successes. So I really, really like that. And obviously it's worked very well for the culture of CTMC, because especially with smaller companies versus, the big, large companies like Teva, then they are very different. They're very two different environments. So I think that's really, really interesting that you've managed to take the best parts of such a large company and really kind of encompass that in your smaller company at CTMC. So yeah, that's awesome.
For sure. There's something about human nature, Rebecca, where when we always want to do better, which is a great, feature, a great aspect that we always why we propel ourselves forward. And so whenever you finish a project or do a lessons learned, there is this human tendency of everyone to be like, okay, what did we mess up? This didn't work as well as we thought. This didn't work. We wanted to do this part better. We always try and be very intentional here about starting with what went right. Because just like we talked about, when you actually there's a lot of ways to do things wrong. There's probably only there's fewer ways to do things right. Once you find some of those that work well, being able to identify those, recognize those, and make sure you keep doing those because you figured out something that works well. Let's make sure, no matter what, that we keep doing those things. And then then not shy away from things that we could have done better and understand what didn't go well, and then iterate an experiment, try something different for those. But then hopefully from that, you keep building on these successes and things that work well.
Yeah, no, I really like that. And I think that's so important. And it's obviously worked really well for you at CTMC. You know, you've got so many things that have gone very right. And from using that method, that's probably one of the reasons why it's worked out so well. One of the things that you've done amazing, obviously, is your partnership with Obsidian Therapeutics, helping them achieve very significant milestones. What would you put down to, aside maybe of, you know, focusing on the successes and how you can replicate that, but what would you say made this collaboration so successful?
There were a number of pieces. So to give a little bit of the background, Obsidian was the first biotech company that we partnered with to take their product, collaborate with them to bring it from research stage through IND enabling studies, through FDA interactions, and into the clinic. And they, at the end of last year, at the end of 2023, reported top-line clinical results, and in 2024 did a Series C raise of $160 million. So the total amount of money they've been able to raise since starting the collaboration with us has been $270 million, and they've reported great results in very advanced stage metastatic melanoma with a novel TIL therapy. So they have done, give them all the credit in the world for coming up with a brilliant therapy that addresses an unmet medical need. And from the beginning, when we interacted with them, we just loved the product. We thought, well, this can really solve a very large problem in for patients. And I think there were several secrets to the success of that project. I think one was a lot of trust early on in the relationship, because the business model that we developed is basically the company puts a lot of trust, a lot of their eggs are in our basket. If we don't deliver, this is typically their first in-human project. They're going to be their first clinical project, and that's sort of their baby, as a biotech company. And so if we drop the ball, it's going to be a big problem for them. And they're asking us to do a very comprehensive partnership in terms of these IND enabling studies, supporting the regulatory interactions, and executing on the clinical study. On the other hand, I think, sometimes there's a false sense of risk mitigation, if you like. I think that's what we talked about with the leadership of Obsidian at the time. Like, well, you could spread all those responsibilities out to 6 different organizations, but if any one of those fails, you're still not going to get to the clinic. So in some ways, I think it's a better strategy to, if you want to say, put all your eggs in one basket, but then you can really work very closely with that partner, and watch those eggs really closely, rather than trying to have them all spread out all over the place. So I think that was my tortured analogies way of saying, there was trust in the relationship, but very much how we structured the partnership allowed us to move very quickly and robustly. So when Obsidian first came to us, they had in mind a more traditional approach for bringing their product into the clinic, using a network of CMOs and CROs, and felt it would take about 24 months to get from the stage they were at into the clinic. And we were fairly new. We had just acquired this new manufacturing facility. We were just putting the group together, but we projected we could do that because we had some efficiencies. We could realize that we could do that in about 18 months. And so that was super exciting that we could save 6 months off of their timeline. And so, that helped drive the deal. Now, in the beginning, in the early days of a partnership, you can sort of quote anytime you want. It's really what you deliver at the end.
Yeah.
And, the teams worked amazingly well together. And this was during the early days of COVID. So, everyone figuring out Zoom and all sorts of other things. The teams worked really well together. And so we didn't deliver that project in 18 months. We actually were able to deliver it to IND in 16 months.
Oh, wow.
And so despite COVID, despite challenges, despite new partnership structure and everything, I think because of that, we were able to deliver that quickly and get Obsidian to these value inflection milestones quicker than they could have gotten through other means.
That's amazing. And I bet they are very grateful that you saved 8 months if they'd gone with either a CRO or CMO. So that's probably exactly what they were looking for. It's rare in a project that people under-deliver on time. So 2 months off your project delivery time is so good. So I bet they were definitely over the moon. So I guess finally here, Jason, obviously, with your experience across various modalities, we've spoken about it at the beginning. Hopefully cell therapy won't take as long as monoclonal antibodies did to really come to their own. What do you think would be the biggest opportunities for innovation in the next decade, and how can you see CTMC playing a big role in this?
I'll enjoy answering this question. So cell therapies have already shown such amazing efficacy, and we're just getting started. They offer such an opportunity for, if you will, like cellular engineering, genetic engineering, to secrete other proteins, to alter the cell surface composition. It's such a vivid platform for engineering a therapeutic, and we're getting better and better tools at doing that engineering, whether it's CRISPR editing or other kinds of tools, synthetic biology. So I think there's tremendous potential in those kind of innovations. With the complexity of having living cells comes this opportunity for engineering them in all sorts of different ways. And what I think CTMC's role in that is going to be, and I think what the key to maturing modality is how rapid is the cycle time from idea to figuring out whether it works well or not, and then iterating that. And so that example I told you about Obsidian is so powerful because, they knew, 8 months earlier that their product was going to work or not. And then not only get that product further through clinical and hopefully to commercialization earlier, but as well, the whole field knows that, what Obsidian did with TILs was very successful. So can we build off of that and make a next generation? And that improving that cycle time is what really helps drive the speed of maturation of a therapeutic modality. And so what we think about a lot is how can we reduce that cycle time and add as much value to each of those cycles as we can. And so whether it's integrating different technology, running innovative clinical studies so we learn the most out of each clinical study as we can, and having just an innovative business model so that we can have more and more shots on goal with these innovative cell therapies. I think that's the role that CTMC can play in helping to mature the cell therapy field.
Amazing. Perfect. I'm very excited to watch how it all unfolds over the next 10 years. I think there's a lot of exciting things to come in the life sciences space. I know the last couple of years have been a little bit tricky, but I think things are really starting to change, and I'm so excited to see where the cell therapy space goes, because I think it's becoming more and more frequent that you get these amazing news articles saying that someone was in stage 4 cancer, 2 weeks left to live, and they've been given this cell therapy and they are completely cancer-free and living an amazing life. And it's just, it really, I mean, I'm, like I say, I'm not a scientist myself. So even though I can only play a very small part, it's just so exciting working in an industry that is really making movements like this.
Rebecca, I think you playing a role in, communicating to a broad audience what's going on. There's going to be this transformation that occurs, because these treatments are just different than historical medicines, because they're single dose. And then the effects can go on for years, if not a complete cure. And so we're starting in cancer, but we're starting to see amazing results in autoimmune disease. People who have been on chronic treatments for years, if not decades, and then given a single dose of cell therapy that resets their immune system and corrects some of these autoimmune diseases. And that's just the beginning. There's going to be so much when we look back on this in 10, 15 years. Our kids are going to be like, what were you guys doing before without these cell therapies and treating chronic diseases just by ongoing daily maintenance of different treatments? It's going to be transformative.
Yeah, absolutely. Well, thank you so much, Jason, for coming onto our podcast. It's been so exciting to talk to you. Definitely really excited to continue following the journey of CTMC, and I hope all our listeners will be doing that as well.
Thanks so much for the discussion, Rebecca. I enjoyed it.
Yeah, thank you so much.